Welcome to the PubMed Daily Review for March 29 SD. Today we scanned 9,528 new open access papers. Here are the top 5 selected for detailed analysis. Up next, our first paper out of 5, Neoantigen-Encoded Oncolytic Viruses as Personalized Cancer Vaccines by Al-Mohanad Al-Khail. Let's break down its key contributions. Cancer vaccines have seen a resurgence, driven by our ability to identify unique tumor mutations called neoantigens. While personalized neoantigen vaccines can generate strong immune responses, they don't always translate to clinical benefit. This is often due to challenges in getting those immune cells into the tumor and overcoming the tumor's defenses. Oncolytic viruses, on the other hand, are engineered to infect and kill cancer cells, while also stimulating an immune response directly within the tumor. This paper reviews the concept of neoantigen-encoded oncolytic viruses as a next-generation cancer vaccine. It explores how these viruses can be engineered to carry specific neoantigens identified from a patient's tumor. The idea is to leverage the virus's ability to infect and destroy cancer cells, while simultaneously delivering personalized vaccine components directly to the tumor site. This integrated approach aims to overcome the limitations of traditional vaccines and virotherapies. The authors highlight several key findings for optimizing these therapies. They emphasize the critical role of CD4+ T-cell help in generating durable anti-tumor immunity, suggesting payload designs that promote this. They also discuss strategies for selecting and encoding neoantigens, such as prioritizing highly clonal mutations and using different payload formats like mini-genes or long antigens. Overcoming pre-existing antiviral immunity is also noted as a significant challenge that needs to be addressed for effective repeat dosing. In conclusion, neoantigen-encoded oncolytic viruses hold significant promise for the future of personalized cancer vaccines. By strategically combining viral infection with patient-specific neoantigens, this approach aims to create a potent in situ vaccination effect. Future work will focus on refining payload design, enhancing immune responses, and developing practical clinical strategies to bring these advanced therapies to patients. Up next, our second paper out of 5, From Genomic Diagnosis to Personalized RNA Medicine, Advances in Next-Generation Sequencing and of Antisense Oligonucleotide Therapies for Rare Genetic Diseases by Paris Rodriguez-Carstens and Tasha Fumiokoa. Let's break down its key contributions. Rare genetic diseases are a significant global health challenge, often leading to long diagnostic odysseys and limited treatment options. However, groundbreaking advances in genomic sequencing and RNA-based therapies are now changing this landscape. This paper explores how these powerful technologies are coming together to offer hope for previously untreatable conditions. The core of this approach involves a two-step process. First, advanced sequencing technologies like WGS and RNA-seq provide a comprehensive genetic and transcriptomic diagnosis. Once a specific genetic defect is identified, customized antisense oligonucleotides, or ASOs, are designed. These ASOs work at the RNA level to either silence harmful transcripts or correct splicing errors, ultimately aiming to restore normal protein function. The results are promising. Comprehensive sequencing has dramatically improved diagnostic accuracy for rare diseases. We've seen landmark N of 1 ASO therapies like Millicin show success in individual patients. Furthermore, the FDA's approval of tofersen for SOD1 ALS marks a significant milestone for ASO therapeutics. While challenges in delivery and access persist, these advancements represent a major leap forward. In conclusion, the synergy between advanced genomic diagnostics and tailored RNA therapeutics is paving the way for a new era in rare disease treatment. While hurdles remain, particularly concerning delivery and accessibility, ongoing innovation in sequencing and ASO design holds immense promise. This integrated approach is fundamentally reshaping the therapeutic landscape for individuals with rare genetic conditions. Up next, our third paper out of 5, Artificial Intelligence-Driven Discovery and Optimization of Antimicrobial Peptides Targeting Escape Pathogens and Multi-Drug-Resistant Fungi by Kalina Wu M.O. and Rafael Maldonado Hernandez from Department of Biology, University of Puerto Rico, Calle Campus, Calle 00737, Puerto Rico, CWM, AFG, JMD. Let's break down its key contributions. Antimicrobial resistance, or AMR, is one of the most pressing global health challenges we face today. Millions of deaths worldwide are linked to drug-resistant infections, and the effectiveness of our current antibiotics is rapidly declining. This crisis is driven by pathogens like the escape bacteria and emerging fungi, which are increasingly difficult to treat. We urgently need new therapeutic strategies, and antimicrobial peptides, or AMPs, are showing great promise as a distinct alternative to conventional drugs. This paper reviews how artificial intelligence, particularly machine learning and deep learning, is revolutionizing the discovery and optimization of antimicrobial peptides. These AI approaches allow researchers to predict peptide activity, selectivity, and even toxicity, moving beyond traditional methods. By learning from vast data sets, AI can rapidly screen potential candidates and even design novel peptide from scratch, significantly accelerating the development process. The results show that AI models, from traditional machine learning to advanced deep learning architectures, are achieving high accuracy in identifying potential antimicrobial peptides. Generative models are particularly powerful, enabling the design of entirely new peptides optimized for multiple objectives simultaneously. This AI-driven approach, often integrated into design test-learn cycles, is dramatically accelerating the journey from initial concept to potentially viable therapeutic candidates. In conclusion, artificial intelligence offers a transformative pathway to combat the growing threat of antimicrobial resistance. By leveraging AI, we can accelerate the discovery and optimization of novel antimicrobial peptides. However, challenges in clinical translation, such as ensuring peptide stability and efficient delivery, still need to be addressed. Moving forward, integrated AI frameworks that incorporate real-world resistance data will be crucial for developing the next generation of effective antimicrobial therapies. Up next, our fourth paper out of 5. Patient-derived brain organoids reveal divergent neuronal activity across subpopulations of autism spectrum disorder by Nissim Peretz and Ariel Tenenbaum from ETI and Beyond Ltd. Jerusalem, Israel. Let's break down its key contributions. Autism spectrum disorder, or ASD, is a complex condition affecting brain development. Its underlying causes are still not fully understood, making it hard to develop effective treatments. Recently, scientists have developed brain organoids derived from patients, which are like mini brains grown in a lab. These organoids allow us to study brain activity and function in a way that was previously impossible, offering new hope for understanding ASD. To investigate ASD, researchers used brain organoids grown from cells of individuals with ASD and healthy controls. They measured the electrical activity of these organoids, looking at both resting activity and how the networks responded to stimuli. Advanced computational methods were used to analyze the complex data, revealing specific patterns of neuronal activity. The study found significant differences in how neurons in ASD organoids behave compared to those from neurotypical controls. This was true for both spontaneous resting activity and for how the neural networks responded when stimulated. Importantly, these differences were not uniform across all ASD cases, but rather showed specific patterns depending on the subtype of ASD. This research highlights the power of brain organoids to model the complex and varied nature of ASD. By revealing subtype-specific electrical activity patterns, these organoids could become valuable tools for developing new diagnostic methods, screening potential drugs, and ultimately, finding better treatments for individuals with autism. Up next, our fifth paper out of 5. Systematic optimization of proteolysis-targeting chimeras for PIN1 enables selective degradation and anti-tumor activity in vivo by Yueying Ma and Zufeng Guo from College of Pharmacy, Chongqing Medical University, Chongqing 400016, China. Let's break down its key contributions. The protein PIN1 is a key player in cancer, often overexpressed and linked to tumor growth and resistance. While inhibiting PIN1 has therapeutic potential, current drugs aren't perfectly optimized. This study explores a different approach, targeted degradation using PROTACs. The goal is to systematically improve these PROTACs to achieve selective PIN1 degradation and see if it works as an anti-cancer strategy. To tackle this, the researchers designed 2 sets of PROTACs, molecules that tag proteins for degradation. They systematically varied the linker connecting the PIN1 binding part to the cell's degradation machinery. They tested these compounds in cancer cells, looking at how well they degraded PIN1. This led them to identify a promising compound, PC2, which uses the CRBN pathway for degradation. The key results were quite encouraging. The lead compound, PC2, demonstrated significant tumor growth suppression in a mouse model. Importantly, this was achieved without any observable toxicity. While its effect on cancer cell growth in lab dishes was modest, its in vivo performance suggests a promising therapeutic window. So what's the big picture? This study provides crucial design rules for creating better PIN-1 targeting PROTACs. It shows that selectively degrading PIN-1 can indeed lead to significant anti-tumor effects in living organisms. PC2 represents a major step forward as the first PIN-1 degrader tested in animals, paving the way for targeted PIN-1 degradation as a viable cancer therapy.